Ischemic reperfusion injury by Ahmadaglin, MD, Alexandria University Critical Care Department.
In the name of Allah, the entirely merciful, the especially merciful.
Now, we arrive at the heart of the problem, the pathogenesis of ischemic reperfusion injury. How does the simple act of restoring blood flow trigger such devastating damage? The answer lies in a perfect storm of interconnected pathological events that are unleashed at the moment of reperfusion. The key players in this destructive cascade are calcium Ca² overload, a massive and uncontrolled influx of calcium into the cells, poisoning them and activating destructive enzymes. Reactive oxygen species ROS, a sudden, explosive burst of toxic free radicals that directly damage cellular structures like lipids, proteins, and DNA. Reactive nitrogen species are NS, closely related to ROS. These molecules, like peroxynitrite, cause nitrosative stress, further amplifying the injury. Hydrogen ion H overload. The built-up acidic environment from ischemia creates a pH paradox upon reperfusion, triggering cell death pathways. Complement system activation. The body's own innate immune system is inappropriately activated by damaged tissue, launching a destructive inflammatory attack. Renin-angiotensin system RAS activation. This key blood pressure regulator becomes dysregulated, promoting vasoconstriction, inflammation, and fibrosis in the reperfused tissue. Individually, each of these factors is damaging. Together, they create a synergistic and catastrophic cascade that leads to the cellular fates of apoptosis, necrosis, and organ dysfunction we've already discussed. To understand the catastrophe of calcium overload, we must first appreciate the exquisite precision of normal calcium handling in a cell. In a healthy cardiomyocyte, the concentration of free calcium in the cytoplasm, known as the in situ matrix calcium, is tightly regulated within a narrow range. As reported, at low pacing rates, this concentration is maintained at a very low level, between 100 and 200 nanomoles per liter. Even during periods of high demand, such as during exercise or stress, the cell manages this increase in a controlled manner. With beta-adrenergic stimulation, which mimics the fight or flight response, the calcium level rises, but only to a range of 500 to 800 nanomoles per liter. This controlled rise and fall is the basis of normal, forceful cardiac contraction. This data establishes our baseline. It shows us the incredibly low resting level and the controlled peaks that a healthy cell can manage. This makes the thousand-fold higher levels seen during calcium overload reaching micromolar or even millimolar concentrations all the more devastating. The cell's normal control mechanisms are completely overwhelmed, turning an essential signal into a potent toxin. Now, let's contrast that precise, controlled physiology with the chaotic reality of ischemic reperfusion injury. During ischemia, the cell's energy reserves are depleted. This cripples the ATP-dependent pumps, especially the sarcoplasmic, endoplasmic reticulum calcium ATPase SERCA, and the sodium-potassium pump Na plus K plus ATPase. The failure of the Na plus K plus pump leads to a rise in intracellular sodium. This, in turn, causes the sodium-calcium exchanger and CX to reverse its direction, pumping sodium out and tragically calcium into the cell. So, even before reperfusion, calcium begins to slowly accumulate in the cytosol. Then, at the moment of reperfusion, the situation becomes catastrophic. With the sudden influx of oxygen and substrates, the senior attempts to resequester the excess calcium but becomes overwhelmed and dysfunctional, leading to spontaneous and massive calcium releases known as calcium sparks. This results in a vicious cycle. The mitochondrial membrane pores open in response to the high calcium, leading to a collapse of the energy-producing membrane potential, which further impairs the cell's ability to pump calcium out. The result is a massive and sustained elevation of cytosolic calcium, rising from the normal nanomolar range to micromolar or even millimolar concentrations. This calcium overload is not a signal anymore. It is a potent activator of necrotic and apoptotic death pathways, directly poisoning the cell. This slide brilliantly outlines the precise biochemical domino effect that occurs during the ischemic period, priming the cell for catastrophic failure upon reperfusion. It all begins with hypoxemia, the lack of oxygen. This forces the cell to switch to an efficient anaerobic glycolysis. A key byproduct of this process is lactic acid, leading to a sharp increase in cytosolic hydrogen ions H, or intracellular acidosis. This acid load activates the cell's emergency response, the plasma membrane Na plus per H plus antiport. This exchanger tries to normalize the pH by pumping H out, but it does so by pumping sodium Na in, causing a dangerous rise in cytosolic sodium. Meanwhile, ATP depletion cripples the cell's primary pumps. The Na plus K plus ATPase fails, meaning the accumulated sodium cannot be removed. This also stops the Na plus Ca squared plus antiporter from effectively pumping calcium out of the cell, as it relies on the sodium gradient established by the Na plus Ek plus pump. The crisis extends to the endoplasmic reticulum, the cell's internal calcium store. The Circa pump, which normally sequesters calcium into the is deactivated. Furthermore, the ryanodine receptor becomes dysfunctional and leaks stored calcium out into the cytosol. The final, toxic result of this entire cascade is a cell flooded with hydrogen, sodium, and calcium ions. This ionic chaos is the unstable foundation upon which the explosive injury of reperfusion will be built. We've seen how calcium overload poisons the cell directly. Now, we see its second, perhaps more devastating, role, activating a powerful inflammatory army that attacks the tissue from within. The increased intracellular calcium doesn't just stay in the cytosol. It triggers the generation of specific danger signals, or DAMPs. Specifically, it leads to the formation of calcium pyrophosphate complexes and uric acid crystals. These substances are recognized by the cell's internal security system, protein complexes called inflammasomes. When calcium-dependent danger signals bind, the inflammasomes activate. This activation is a major alarm bell. It leads to the massive production of potent pro-inflammatory cytokines, most notably interleukin-1 beta, IL-1 beta, and tumor necrosis factor alpha TNF. These cytokines then activate master transcription factors like nuclear factor kappa B, NFP, which in turn switches on the genes for even more cytokines and chemokines. The result is a self-amplifying, destructive feedback loop, a full-blown cytokine storm within the reperfused tissue. This storm recruits inflammatory cells, damages the microvasculature, and kills neighboring cells, thereby greatly exacerbating the initial IWR injury. In essence, calcium overload is the match that lights the inflammatory fire, transforming a local injury into a widespread inflammatory assault. Beyond sparking an inflammatory fire, the elevated cytosolic calcium also acts as a direct executioner within the cell, activating a suite of destructive enzymes that dismantle the cell from the inside out, steering it decisively toward apoptosis. First, it activates calcium-dependent phospholipase A2. This enzyme attacks the very fabric of the cell, its membrane. It breaks down phospholipids, damaging the integrity of the plasma membrane and the membranes of vital organelles like the mitochondria. Second, it activates endonucleases. These enzymes are like molecular scissors for the cell's genetic blueprint. They enter the nucleus and cleave DNA, both non-specifically, causing chaos, and specifically, creating the characteristic DNA laddering that is a hallmark of apoptosis. This makes cell death irreversible. And third, it activates proteases, most notably calpins. These enzymes degrade a wide range of essential cellular proteins, including structural proteins in the cytoskeleton and key enzymes, leading to a complete breakdown of cellular architecture and function. The simultaneous activation of phospholipase A2, endonucleases, and proteases represents a coordinated, calcium-triggered demolition of the cell structure, its genetic material, and its functional machinery. This is the biochemical definition of the programmed cell death we see in IWR.
Thank you.